understand this, now we will see some animations which are helpful to understand the concept of the enzyme reversible enzyme inhibitors. Okay, so now we will see the animation which is regarding with this enzymatic inhibition. So wait a minute, I will share you.
Hi there, and welcome back to our
The
inhibition
inhibition.
on understanding
Now
medications. In
you
this
can
lesson,
see
we're going to be talking about another one of the general ways in which our medication,
where we are going to see the competitive inhibition and what is the non-competitive inhibition and how these are going to happen with the enzymes, right? So now we will start with this animation. So now that animation is in front of you, is it visible now? Is it
Yes, sir.
visible? Okay, so now we'll discuss about this enzyme inhibition.
enzymes are proteins that catalyze, or in other words, speed up a reaction. So in the body, there may be a reaction that occurs, but it may take maybe 3 hours. And with the enzyme, it may speed up that reaction, and the reaction may occur in a few milliseconds or a few thousandths of a second. Now, this is depicted in the really simplistic diagram here. The purple is the enzyme, and the smaller structure in red is the substrate. A substrate means that it is the substance upon which the enzyme is acting. And as they come into contact, these two molecular structures have kind of a bond, a molecular bond, which allows them to bond and allows the reaction to occur. And as that happens, there's going to be a cleaving of the substrate into two different.
So here we can understand this purple one is the enzyme to which the substrate binds, and due to the enzyme catalytic activity, they are going to forming the products, right? So they are going to form a product.
Products. Enzymes are really important to a range of biological processes. So given that, it's understandable why a lot of our medications work by inhibiting one of them. So let's try to understand the basics of enzymatic inhibition as it applies to our medications. When you think of enzymatic inhibition, you should immediately think of two major types of enzymatic inhibitors, the reversible inhibitors and the irreversible inhibitors. By far the largest group of enzymatic inhibitors is the reversible inhibitors. And those are divided into at least 4 subsets. Clinically speaking, the most important group of reversible inhibitors is the competitive inhibitors. But there is another type that's called non-competitive inhibitors, and we should know a bit about them because they're oftentimes misrepresented by authors as being an irreversible inhibitor, and they're not. Remember that both the competitive and the non-competitive inhibitors are reversible. In competitive inhibition, the medication binds to the active site of the enzyme and prevents the binding of the substrate. So it is competing with the substrate. Because it is a competition between the medication and the substrate, adding more substrate would normally increase the amount of binding of the substrate. That is the largest group of reversible enzymatic inhibitors, the competitive inhibitors, and when the medication is fully metabolized, there should be no further effects. Now let's take an example of competitive inhibition to make sure that you understand that. The structure and purpose.
So same example we are taking the acetylcholine esterase or malonate.
Enzyme. Let's use the example acetylcholine esterase, the enzyme that breaks down the neurotransmitter acetylcholine. The structure in red is the substrate, and in this case, the substrate is acetylcholine. When the enzymatic action happens, acetylcholine will degrade to acetate and choline. Each molecule of acetylcholine esterase degrades about 25,000 molecules of acetylcholine per second. And finally, the medication is in yellow. The medication is competing for the active site on the enzyme, meaning that every second that it is in the active site is a second that the enzyme is not degrading its 25,000 molecules of acetylcholine. The other major type of reversible inhibitors that is fairly common is a non-competitive inhibitor. In that case,
what happens in non-competitive inhibition, as we learn in these concepts? What happens? Where the inhibitor is going to bind in the non-competitive inhibition? Yes, anybody? Where the inhibitor binds in non-competitive inhibition? Yes, Renuka?
Allosteric site.
No. Seema? Sapna? Divya? Mahesh? Where the inhibitor binds in non-competitive inhibition type of reactions? Fatafat. Anybody?
the inhibitor binds to what we call an allosteric site.
Okay, Renuka. But my question is the enzyme substrate complex को bind होना चाहिए था वो, ठीक है?
It's not the site that's in the active portion of the enzyme, but in a different portion of the enzyme. This diagram denotes a non-competitive inhibitor changing the active site so that the substrate cannot bind. Now let's talk about irreversible inhibitors. If a person is given an irreversible inhibitor of an enzyme, the enzyme can no longer perform its function. An irreversible inhibitor inactivates an enzyme by binding normally covalently to the enzyme and normally at the active site. And you may be thinking right now, what are we going to do without that enzyme? And the answer is just because it is an irreversible inhibition, it doesn't mean that we'll never have that enzyme again. We just need to make more of that enzyme. So from a clinical aspect, you can understand how an irreversible inhibitor is often going to last a lot longer than the effects of a reversible inhibitor. An example is the proton pump inhibitors, such as omeprazole and other medications that end in the suffix prazole. Those have a relatively short half-life of about 1.5 hours. And that would normally mean that the medication...
So this prazole tablet which we are going to take for as a antacid. So whenever the pentapanozole, omeprazole, omez D या पैंडी लेते हो तो वहां पर होता है prazole. So that is going to inhibit the proton pump. So it will going to inhibit the H plus, high amount H plus in the intestine, so we can feel that we are having the acidity. So it is going to inhibit that pump. So H plus ions, which is a protons, are not going to come into the intestine. So we can say that there will be the suppression of that particular proton pump.
Would have to be taken quite frequently. But because it is an irreversible inhibitor, our parietal cells in our stomach need to make more enzyme, and that can take up to 72 hours to fully replenish the enzymes. So omeprazole and other irreversible proton pump inhibitors
Yes, Renuka.
taken on a
सर मग इनोची पण सेम कन्सेप्ट आहे का?
इनो कंटेंट में अगर प्रेजोल है तो हो सकता है, मे बी. ओके, सो कंटेंट आपको देखना होगा वहां पर. सो पैंडी का मुझे पता था, ओमेस का पता था, इसलिए मैंने बोल दिया कि whenever the suffix prazole is there, so it will going to inhibit the proton pump. Okay?
Yes, sir.
daily basis. Irreversible enzymatic inhibition is also important in toxins such as pesticides and anti-infective agents. For instance, many of the nerve gases are irreversible inhibitors of the enzyme that breaks down the neurotransmitter acetylcholine. And acetylcholine works all the way through the body. But let's just take the example of what happens at the site of the muscles. Acetylcholine is the neurotransmitter that's responsible to instruct the muscles to contract. Without the enzyme that breaks down the acetylcholine at the muscle, the acetylcholine continues to stimulate, for instance, the respiratory muscles. And remember what we need in order to breathe. We need to be able to contract the diaphragm, but we need to be able to relax it as well. So contraction, relaxation, contraction, relaxation. And with this nerve gas that's an irreversible inhibitor of that enzyme, it means that there's constant excesses in acetylcholine and constant contraction. So the person succumbs to respiratory distress. We also use irreversible inhibitors when we're targeting infective agents. For instance, penicillin is an irreversible inhibitor of an enzyme which only the bacteria have, but humans don't have.